Hi, I'm Professor Brittany Gill, and let's get started. Today we're going to be learning about Chapter 17, Evolution of Microbial Life. We're going to be talking about viruses. We're going to be talking about general information, the general structures, host specificity, replication, diseases, and treatments. Then we're going to talk about prokaryotes, the general information involved with them, their structures, functions, how they reproduce, a couple different modes of nutrition, as well as disease and treatment. Then we're going to get a little bit specific about two of the domains we've learned about, domain bacteria, and then we're going to finish off talking about domain archaea. Viruses. So, are viruses alive? Think about that for a minute. How I like to classify them, I call them the living dead. The reason for this is they aren't alive. They do not meet the qualifications of life. Why, you might say? Because they are non-cellular. Therefore, they are non-living. They're acellular or particles. They're not in any domain and any kingdom. We also, as we go on through this chapter, we're going to learn a couple reasons other reasons why they're not considered living. Viruses are smaller than bacteria. They're approximately 0.2 to 2 micrometers. And to give you that estimate, 1 micrometer is 1/1,000,000th of a meter. So they're tiny, tiny. So a meter is about 3.3 feet, so about a little bit over a yard. So viruses are very diverse in structure. However, they all are composed of two distinct parts. The first one is that they all have what's called an outer protein capsid. They also have an inner nucleic acid core. This could either be DNA or RNA. Some viruses that inflict animals also have an envelope. So some animal viruses have 3 parts, but every virus has at least those 2 body parts, the protein capsid and the inner nucleic acid core. Viruses are also categorized by what type of nucleic acid they possess, their size and their shape, meaning if they're single or double-stranded, and also the presence or absence of that outer envelope. Now here are 2 pictures of an adenovirus, which we call a naked virus, because it does not have that outer envelope. It just, you can see in this picture here, has the outer protein capsid, this green area, and then it has, in this case, that RNA, that nucleic acid core. Now, on an animal virus, you can see here that it's surrounded by an envelope, and they generally have spikes. These spikes are sensors that allow them to attach or to adhere to different cells of the animal. So you do see this envelope here, and then you can see in here, it looks almost identical to this one. They have that capsid and then the nucleic acid core. Now, all viruses have what we call a specific host range, and we're going to be talking about a couple of those. But I want to talk about first are the bacterial viruses. Bacterial viruses are called bacteriophages, or also just phages. We also have plant viruses and animal viruses. So, but looking at this picture here, this is a bacteriophage. This is what's going to inflict bacteria. You can see it has the capsid head and the inner nucleic core here. But you can tell it has this extra part here. So this tail sheath is what it's called, and these kind of little pins and legs to help them attach to the bacteria, so that they do not have an envelope, but they do have this other body part down here of the bacteriophage. Looking at these pictures here, this is an example of a plant virus. This is that naked virus here, the adenovirus. Here is an influenza virus, so what we have, you can tell it has that animal envelope here to connect to our cells. And then this is a picture of a bacteriophage. So here's a real scanning electron microscope view of these different viruses. So as we talked about, since viruses are not alive, they are not classified using the Linnaean system. But they do have the capability to mutate or adapt/evolve, if you will, and multiply. So they do seem to have things that are characteristic of living organisms. That's why I kind of call them the living dead, because they still do things that make them seem alive, but they're not. And one of the other main reasons, besides not having cells, is because they can only replicate within another living cell. So they are considered what we call obligate intracellular parasites. So they essentially take over a host cell, so a bacteria in this case, and they use the bacteria's form of machinery for replication. So they steal another living organism's machinery. Now, replication processes within, we're just talking about bacteria, viruses, okay, so bacteriophage. We have the lytic and the lysogenic cycles. The lytic is shorter, it only has 5 letters. So compared to the two, that's one way to remember. The lytic is shorter. Lytic means to loosen or to break, like lysis and lytic are very similar words. So this is the process of the lytic cycle. You have attachment, penetration, biosynthesis, maturation, and release. You do need to know this order. The lysogenic cycle is longer, and what makes it longer is because it has this dormancy or latency period. It's very similar, except for it has that time when it doesn't actually do anything. So you have attachment, penetration, and then here's the new step, integration, which we call a prophage stage. Then you have biosynthesis, maturation, and release. So it just has that one extra step between the differences, but it makes all the difference. So let's look a little bit more specific here. We call it lock and key receptors, meaning specificity. So they actually have special places that they latch on to the bacteria, and it allows them to know exactly where they need to essentially penetrate into that cell. So when a virus penetrates into a bacteria, we talked about the outer protein capsid, that does not go in. Only the internucleic acid cores, be it DNA and RNA. That's the only thing that's actually going to be entering into the host cell. Now, right here on the lysogenic cycle, this integration part is when viruses are not actively being replicated. The DNA is incorporated, but they're not making any viruses. And then in release, this is when cells, what we call lysis, and so they break. They actually, once the viruses are being produced inside the cell, when the cell has made enough, I guess, it will rupture and kill the cell, and the viruses will escape. So watching this animation here, we see the attachment, and now we see the penetration, and it's just the DNA or RNA coming in. Once that happens, replication occurs. Then, once there's enough, they will release. So that's what this video is kind of showing you here. So remember, absorption is the first thing that's going to happen, okay? Once the penetration has occurred, maturation is going to develop. As you can see here, it's going to use this machinery of the bacteria to create more viruses. And then, as you can tell at the very end, this poor bacteria dies, the cell will rupture, and all the little new viruses will come out. So this results in the death of the host cell. So lytic, once it attaches, it's the end of that bacteria. It's no longer going to be able to survive once the process of maturation occurs for the viruses. We call this a virulent virus. Now this only occurs during the lytic cycle, that the host that it is in will actually die. Now let's look at the lysogenic cycle. The first steps are just like the lytic. However, in this cycle, the host is not destroyed by the virus. Because what happens is that the nucleic acid, so that DNA or RNA, whatever comes in, is actually going to join the cell's DNA. So the bacteria DNA will incorporate some of that viral DNA. And we call that a prophage now. So bacteria DNA is actually circular. Ours is way more complex, but theirs is circular here. So what happens is you have that penetration, or the attachment, penetration. Here's what it comes in, okay? And that's when we get this prophage. It comes in, and here's the green is representative of the bacteria DNA. That, I guess, pink is the new or the viral DNA. So once that's incorporated into the bacteria cell, it just stays in there for a little while. And you can see that that cell it can reproduce and make more cells, more bacterium. Now this new bacterial cells actually have bits of copied of the replicated viral DNA. So you can have one bacteria cell that once it replicates, you're going to have hundreds, thousands, depending on how long the viral DNA is, I guess, dormant before it actually is replicated. So you'll have many cells that will reproduce through the normal cell binary fission and replicate, and you'll have that viral DNA incorporated in it. This could go on for years, and we call this a temperate virus because it's capable of using the lytic and then, well, it has a lysogenic cycle, and eventually the lytic cycle will undergo itself. So this is how we have HIV, this is how cold sores come up, or shingles. And a lot of diseases that we get, they say you have it for life. It's because once you have that DNA, that your DNA has now incorporated, let's say the new viral DNA, you will always have that in your system. And so right here, at first, it does not kill the host cell. It will just allow the cell to do its normal life cycle, reproduce and create more bacteria cells, and now have the new viral cell, or excuse me, more viral DNA. So if you look at the cycles together, the lytic cycle will go in and automatically reproduce and make more viral cells, or not cells because they're viral, more viruses, in this case bacteriophages, and they will rupture or lysis the bacterial cell and kill it. And then they'll go off and infect more cells. Now what will happen with the lysogenic cycle, like we talked about, it stays in that dormancy or latent period until sometimes some cue is given to the bacteria cells to say, okay, replicate viral cells. And so then they will go into the lytic cycle, essentially, and once that occurs, that bacterial cell will die, and this is how we get those symptoms to reoccur, like in our bodies. But we're focusing on the bacteriophage right now. Let's look into the replication process within animal cells now. So if DNA is the base, what will happen is you have attachment, penetration, biosynthesis, maturation, and release. So very similar to what happens in a bacteriophage. If it's an RNA-based virus, then you're going to have attachment, penetration, and here's the new steps. Reverse transcription, integration, which is called a provirus here, biosynthesis, maturation, and release. So this is what this penetration here is going to be very similar to what we call a retrovirus like HIV. This penetration, once again, we're going to remove that envelope and remove the outer protein capsid. So only the DNA or the RNA will enter into the animal cell. So that's something to remember. Now when it's released, what's neat about this is because all viral cells that are inflicting animal cells have that outer envelope. And so what happens is you get this budding effect. So those viruses, once they've developed, they go to the outer edge of our animal cells and they stick to the side, and we call it budding, as it forms this envelope on the outside, and then it's able to bud off and have a new virus. So here's a picture to help you kind of see that. So starting here, you have attachment, and it's the spike combines with the receptor. So because all our cells, they're not just a big bubble. They have a lot of different things on the outside. And so when those viruses come, they kind of trick the cell and attach to the certain places, and they allow the information of the virus to come in. So that's when you have penetration. Now only the DNA in this case, because we're talking about the DNA part, will come in, and that will get incorporated. It will go into the nucleus. Biosynthesis will occur, and so you'll have protein synthesized. You're going to have DNA produced. Once those proteins are synthesized and the new strands of DNA have been put together, these viral cells, or I keep saying cells, these viruses will come in and start producing and building new ones. And you can see over here, here's the budding effect going on. So once you have the inner parts of the virus, they'll go to the outer membrane and be able to form a new layer of the envelope. And that's how the budding works. Now, let's look at what happens if it's RNA. So this could be, for example, an HIV virus. So what happens is you see up here, number 1, the attachment, same kind of deal, penetration. Now what happens here is reverse transcription. So we'll learn a little bit later, but in also in Bio 114, you learn transcription and translation and how that all works with the RNA. And so in here, we have to make it essentially a form of DNA. And so there's just the extra steps that the DNA can be incorporated into the human, or the human host, in this case for the HIV, and then it's a part of that DNA. Once it's a part of the DNA, then biosynthesis occurs, and then maturation, and so then proteins will be developed, and the different parts of the virus will be formed, then it'll mature, put together kind of like building blocks, I don't know, and then it will be released out into the cytoplasm and out into the environment of the host cell. And so what happens here is there's just that extra step, because RNA is not the same as DNA, so you have to convert RNA into DNA so that the cell can replicate it. Let's look at some examples of diseases. The first one we'll want to go into are some plant viruses. We have the tobacco mosaic virus, that's what this is, TMV. Banana streak virus, you have carrot thin leaf viruses, so lots of plants are affected by viruses. And have huge economic repercussions. Same thing with animal viruses. We have rabies, polio, mumps, chickenpox, smallpox, and influenza. Here's an example of mumps. And so this, many of you have probably already had chickenpox, or at least have had the vaccination for it. Your dogs get rabies shots, your cats, every year or so. Polio, that still exists in the world. Both of my grandparents have polio, or had it, they still do. And so those things are because of the animal viruses. Now we have something called the emerging viruses. These are the big ones that are starting to come in today's life. So a couple years ago, I don't know if you remember, but the Ebola virus was a big deal, and coming in to the United States. We have the avian influenza, SARS, hantavirus, West Nile. All of those are some viruses that are starting to emerge into today's society. Let's talk about treatment and prevention. So treatment are antivirals, and I want to make sure you guys know that it's not antibiotics, because these are viruses, so antibiotics don't work. Those are for bacteria. And so the only thing that we have are antivirals, which are kind of just pills you take to help you prevent to get any of these viral diseases. Other preventatives are vaccinations. That's the number one way to make sure you don't get any of these viral diseases. We have public awareness and vector control. So vector control are anything that could potentially carry the viral diseases. So hantavirus, getting rid of mice in your house, making sure you have no poop and that kind of stuff around. So those are things that we can do to prevent those diseases coming into our homes. Viruses and cancer. Now, there's the human papillomavirus, HPV, and that has increased the chances of cervical cancer if you do have this. Same with hepatitis C. If you have that, your likelihood of getting liver cancer also increases. Alrighty, prokaryotes. Remember, pro means before, karyo means the nut, so before the nut, meaning before they have the nucleus. So these guys do not have a nucleus, they do not have various organelles, okay? Because none of them are membrane bound. Like I said in the previous chapter, they do have ribosomes to help them in their protein synthesis, but that's pretty much it. There's two domains we've talked about that are prokaryotic. They have bacteria and archaea. Now these guys are widely distributed in nature. They pretty much can be found everywhere, the water, soil, air, everywhere the surface is. They are the most abundant of any living organism on Earth. So prokaryotes, as we know, are indeed alive. So we have viruses that aren't really alive, and the next step up are the prokaryotes. And these guys, for example, you have 1 gram of soil, so about a paperclip weight-wise, equals 100 million bacterial cells. So just a tiny amount, tons of these organisms are alive. So all of these are tiny. They're unicellular organisms. They're about 1 micrometer to about 10. So that's, like I said, 1 in 1 millionth. So they are bigger than viruses, but they are still really small. Now, a lot of the structures that are associated with bacteria, you will need to know these. So make sure you memorize these and know where they are on a chart of a picture of a bacteria. So let's go through them and then we can quiz you about that. So you have the nucleoid. This is not the same as the nucleus, but it is the area that contains a singular strand of DNA. Because it's bacteria, like we talked about earlier, their DNA is in a ring. And so they have that circular strand of DNA called the nucleoid. Then you have the plasmid. This is the accessory ring of DNA. And this is what scientists use to do all the cool genetic engineering because since it is in a ring, it's so easy to find. Ribosomes are responsible for protein synthesis. So they make all the proteins in the body of this little cell. The plasma membrane. This acts as a barrier between the internal and external environment. Now I'm going to ask you this on the test, on the quizzes. It is not for protection. It is a thin layer. Think of it as kind of like a balloon. Something can just go in and pop it and it's gone. So the plasma membrane is only a barrier between the internal and exterior environment. So it's kind of like a bouncer. Only certain things can come in, certain things can leave, but it doesn't really do anything as far as protection. Now, cell wall, that can surround the plasma membrane, and this is composed of the peptidoglycan, and this is what is actually used to target, what antibiotics used to target. If you can ruin the peptidoglycan, then you can kill off a bacteria cell. But the cell wall is part of the protection process. The capsule or the slime layer is also something that surrounds the cell wall, and that is protective. And then you have something called the pili. This is a short hair-like structure, and it's used to adhere to different surfaces. And then you have the flagellum. This is the whip-like projection, and this is how it moves. It kind of uses it to swim through whatever surface it's in. Now, these three up here, the nucleoid, the plasmid, and the ribosomes, all are in the cytoplasm of the cell. This is called the cell envelope. This contains the plasma membrane, the cell wall, and the capsule or the slime layer. And then these are the appendages, what it's used on the external outside to interact with the environment. Okay, so I want you guys to pause the video here in a second, see if you can fill in the blanks here and match where the different parts of the different structures of the bacteria cell go. So pause. Okay, pretend you paused. Alright, so these are your answers. Right here, this area is the nucleoid. Down here is the flagella, the big whip-like structure, helps it to move. These are the pili and the external surfaces. In here you see those little dots, those are the ribosomes. And then over here you have the capsule, the cell wall, and the membrane here, cytoplasmic, so it's a plasma membrane. So that's what's going on here. Okay, so reproduction of bacterial cells or prokaryotic cells. They are asexual. As we talked about before, this means without sex. So they have what we call binary fission. This is when you have all the daughter cells are genetically identical to the parent. Now, so here is a picture. Here's the parent cell. When it's undergoing binary fission, it actually will duplicate everything inside and swell. So it is bigger. And then it essentially cuts in half. And so now you have what we call two daughter cells. Even though you start with one cell and you're going to end with two. You're not going to have three. It's not going to give birth to two more. It just cuts in half essentially. So binary, remember bi meaning two. So you have one cell, it expands, and then it pinches off, and now you end up with two daughter cells. And these daughter cells are the exact replicas of each other. They're like clones. And so that is what's going on with binary fission. DNA variations can occur via what we call genetic recombination. So this is not binary fission. This is how bacteria and archaea can actually get new DNA. So we have three different ways. The first one is transformation. This incorporates DNA from the environment into the genome. So there's just scraps of DNA floating around and they pick it up and now it's part of their DNA sequence. Then you have conjugation. This is the transfer of genetic information from one cell to another, what we call a sex pili. It's very similar to just the normal pili. It's also called the conjugation pilus. It's just generally a little bit larger pili that can attach to another cell. The last one we call is transduction. This is when that bacteriophage, so this is specifically for bacteria, remember the bacteriophage, the virus, that transfers bacterial DNA from one cell to another. So these three methods are ways that bacteria can actually develop antibiotic resistance because they have different genetic information, which leads to our antibiotics not destroying this specific, let's say, peptidoglycan of the bacteria. So here's what transformation looks like. You have something of the donor cell, its lysis, it breaks open, and now you have, I guess, floaters of DNA going around. Now what can happen is that those DNAs can get inside the recipient cell and actually be taken up into that DNA. The conjugation, so you have your donor cell, your recipient cell. What happens is maybe there's some other plasmid in there or duplicated or something that's a little bit different, and then that sex pili will come and it will actually give part of that DNA or even the whole plasmid into the other cell. And so that's one way how they can transfer genetic information. The transduction, we've already talked about how the bacteriophage works, how that incorporates, it takes in the DNA, it makes more viruses, and then when they come out, it can actually have some bits of the remnant RNA or DNA of the previous cell, and when it's injected or infects essentially a new cell, that can incorporate some of that new DNA. So that's another way new DNA, RNA can be transferred to a different cell. Here's a really funny comic I saw. So it was on a shortcut through the hospital kitchens that Albert was first approached by a member of the antibiotic resistance. Psst, hey kid, want to be a superbug? Stick some of this in your genome. Even penicillin won't be able to harm you. Now you know how that works. Endospores. Endospores are formed by some bacteria when they're exposed to hostile environments. Have you guys ever heard of anthrax or botulism? This is what they are. So you have your bacteria, and when they're stressed, they create this outer coat, this endospore. So the whole purpose of this endospore is for survival. It's not for reproduction. And so these endospores can last for a long, long time. They can be in soils, they can be in your cans of food or whatever, and they can just stay there. And then when they become active, because of something friendly in the environment, some conditions are just right, then the bacteria can become active again. And that's when it produces those toxins. So you have anthrax and botulism. These will generate endospores. And those toxins are released that cause those different diseases. So over here is a picture of anthrax and its endospore. So like it says here, an endospore's protective multi-layered coat helps it survive in the soil for many years. So that's why it's hard to get rid of. They can last for generations. I think you can find endospores that are thousands of years old, if not older. Let's talk about the means of nutrition. So environmental conditions required for metabolism, which we know metabolism and homeostasis is part of being alive. So metabolism specifically, we're going to talk about cellular respiration, and how that occurs can vary between these different groups. So we have what's called obligate anaerobes. These survive only when free oxygen is not present. So they can't survive when oxygen is around. Anaerobes is kind of not really Greek or anything, but it means without oxygen. So they can only be in an area where there's no oxygen. Then you have something called facultative anaerobes. These grow either in the presence or absence of free oxygen, so they can survive with or without oxygen just fine. And lastly, you have aerobes, and these require oxygen to be present. So think of anaerobic exercise and aerobic exercise. Aerobes require the oxygen to live. Now, modes of nutrition also vary between different groups. We have autotrophs, which we've discussed before, We have photosynthesizers. Specifically in bacteria, we have cyanobacteria. They are critical, so make sure you guys remember them. You also have chemiosynthesizers, and these are the archaeobacteria, so the really old bacteria, I guess, almost archaic is the word for old. They're very similar to archaea, but not quite. We also have heterotrophs. Now, most bacteria are actually going to be heterotrophs, and most bacteria are free living, meaning they don't have to live inside a host. So most bacteria is just out and about, not infecting anybody. There's a lot of ecological and environmental importance. For example, saprotrophs or decomposers, they live off all the dead organic material. So without them, the world would literally stink. And so it's a good thing we have them around. They also are really important for recycling different nutrients into the ecosystem. Because once they're taking up, especially vitamins and minerals, when the dead whatever is broken down, it's able to be absorbed by somebody else in the circle of life kind of deal. They also have a huge economic importance. They're used to make different alcohols, acetic acid, acetones, and antibiotics. They are also used to make food, such as cheese, butter, and sauerkraut. So they're really very important for our lives. Now, some of them have developed what we call symbiotic relationships. Now, I'm just going to put my face here. The first one is called mutualism. This is when both the bacteria and the host can benefit. So a lot of nitrogen fixing bacteria and roots. Now, plants have to have nitrogen in order to survive, and so having that bacteria down there to help fix it or help get it usable for them is very critical. Commensalism. Now, this is when bacteria benefits and the host is not harmed or benefited. So the bacteria gets something, the host it doesn't matter. Nothing's important. Nothing's hurt, nothing's gained. On the other hand, of parasitism, this is when bacteria actually causes harm to the host. So this is the diseases that we're really familiar with. The pathogens is what we call these, those that actually cause harm. So some different examples of bacterial diseases are all the STDs, such as syphilis and gonorrhea. There's a lot of respiratory diseases, like strep throat. Many of you have probably had this before, scarlet fever, tuberculosis. Skin diseases, such as a lot of boils and pimples, can also be caused by bacteria. There's a lot of digestive tract diseases, food poisoning, dysentery, cholera. In the nervous system, we have the botulism and tetanus, leprosy, systemic diseases such as the plague, like bubonic plague, you have typhoid fever, and there's many, many others. It's like Lyme's disease given by ticks. And so the best treatment and prevention. So treatment would be antibiotics. These inhibit the cell wall, and specifically the peptidoglycan, from being developed from synthesis, or it inhibits different protein synthesis. It targets prokaryotic cells and not eukaryotic cells. So antibiotics can differentiate between that because of the different chemicals and how we know what the structure is of the bacterium. Obviously, public awareness. Having good food prep, washing your hands, making sure everything's cooked to the right degree. All those things are really important to make sure that we keep clean. Also, another thing is vector controls. So fleas and ticks, mice, anything else that are out there that could carry things, like the bubonic plague. We get rid of those, keep everything clean, and we should be fine. So let's get into a little bit more detail specifically about domain bacteria. Bacteria is the most common type of all the prokaryotes. There are three basic shapes, and you need to know these. The first is bacillus, which is rod. You have coccus, which is round, and spirillum, which is curved or spiral or helical. How I kind of remember this is that bacillus has two long L's. Technically, is the line here for B, and the two L's, so that's rod. Coccus, it's a lot of circles. If you kind of just draw things over, it's round. And spirillum, it's spiral. It's helical. That's how I remember the three different types. Let's talk now about cyanobacteria. Cyanobacteria can actually be red, yellow, brown. We mostly see it as green, but it can also be those colors because it is photosynthetic. So it has different types of pigments that allow it to make its own food, so it's autotrophic. Now, these guys live mostly in moist environments or in the aquatic world, and they, like I said, they use photosynthesis. Now, the difference between them and a plant, besides being prokaryotic and eukaryotic, is that the thylakoids is where they undergo their photosynthesis and not in the chloroplasts. So it's a little bit different there. Now, one of the really most important things about cyanobacteria is that they actually have thought to be the first organisms to release free oxygen in the Earth's atmosphere. So they're one of the earliest ones that allowed oxygen to be on our planet. These guys may live singly or in colonies or be what's called filamentous. So if it's, for example, a unicellular form, like right here would be the Glyocapsa, how it's just kind of by itself. You have the colonial form, which is Anabaena. So each of these are like unicellular, but they form in this group here. And then we have something called the filamentous, which is the Oscillataria, which is up here. It looks like it's in a tube, and they're highly coiled around each other. And so they can also live in that kind of colony. Now, bacteria lack any visible means of locomotion. Some have what's called the heterocysts that fix the atmospheric nitrogen, like the Anabaena. So those are really, really important here. First, in the aquatic food chain. So the cyanobacteria really are the bottom of the food chain. Everything eats them. They make their own food, and so whatever they take in is from the environment, and then everything eats them. What's also really interesting about cyanobacteria is that they're also thought to be the first colonizers to land. And so these guys have made leaps and bounds in creating the Earth as it is today. Cyanobacteria can also form symbiotic relationships. So with fungi, it's called lichens. And this is important for soil formation. Let me move my picture. But right here is an example of lichen. So it's a fungus plus a cyanobacteria. So they're able to break down the rocks here and use the different nitrogen or whatever minerals are in there. And as it decomposes, it actually makes soil. Some of these cyanobacteria can also cause blooms or fish kills. Now, this isn't the same as an algal bloom, that's a protist, but it's kind of the same idea of how they can make the water toxic and create even lesions on the fish. And so this can also be a bad thing that happens with the cyanobacteria. Alright, let's talk about domain archaea. The composition of the cell wall is different than any other types of bacteria. So that's why these guys are separate in their own domain. They also have very different ribosomal RNA and nucleotide-based sequencing than the bacteria. They share more similarities with domain eukarya than any other bacterial type. And their plasma membrane has a high lipid or fat content. Most are autotrophic by chemiosynthesis, so instead of using sunlight, they use chemicals and the reactions that happen in their environment to create their own nutrition. These prokaryotes also live in extreme environments. They're called extremophiles, meaning they love extreme things. And so they actually have been thought to be one of the earlier primitive organisms on the Earth. So they like to be in the primitive Earth environments. And one of the best things about them is there's none known to be parasitic. So let's talk about some examples. We have the methanogens. So these guys produce methane gas. So they live in the GI tract of animals, they live in the bottom of swamps and marshes, so sometimes that buildup of methane comes up and it bubbles out. That's due to the archaea. Cows, yes, they produce methane, so anything that comes out that might be smelly, that is from partial of the archaea. Now you have the halophiles. These guys live in very, very salty environments. About 12 to 15% salt is such as the Great Salt Lake and the Dead Sea versus the 3.5% that's in the ocean. So they'll really enjoy that really salty environment that many things can't survive in. And so those are the halophiles. You have the thermophiles. These guys love acid conditions and also extreme heat. So you can find them in the bottom of the ocean, in geysers, or in like Yellowstone, and hot springs and thermal vents, like this one down here. And so hopefully this has been helpful for you guys. Let me know if you have any questions, and good luck.